G protein coupled receptor is an example for membrane integral receptor or cell surface receptor. It is a single polypeptide chain. It is made up of a single polypeptide chain. It spans across the cell membrane 7 times, which means we can see that the N terminal is outside the cell surface and the C terminal is inside the cytoplasm. Hence, it is a polypeptide chain and it spans through the cell membrane 7 times. It spans across the cell membrane 7 times. Hence, it is called as 7 transmembrane receptor or serpentine receptor. We can see that the C terminal, which is in the cytoplasm, is attached to a heterotrimeric G protein, which has 3 domains. The 3 domains are alpha, beta, and gamma. In inactive state, the alpha domain is attached with GDP. We can see that the alpha domain is attached with the GDP. When a signaling molecule, a hormone or a ligand binds or comes to the receptor, the C terminal undergoes conformational changes. And thus, it releases GDP and GTP comes and bind there. Hence, the dissociation of alpha from the G protein takes place, and this activates the G protein. The alpha binds to the adenyl cyclase enzyme, which converts cyclic AMP from ATP. Hence, when the adenyl cyclase enzyme is bounded by the alpha domain of the G protein, it converts, it forms or produces the cyclic AMP from the ATP. The cyclic AMP is known as a secondary messenger, and here the primary messenger is the ligand or the hormone which comes and binds to the receptor. The cyclic AMP further activates pKa, which is known as protein kinase A, whose primary role or activity is the addition of the phosphate group. The pKa is a tetrameric molecule which has 2 regulatory subunits and 2 catalytic subunits. The CAMP binds the regulatory subunit of the pKa and dissociated with the catalytic subunit. Now the regulatory subunit goes to the nucleus. Inside the nucleus, it binds with the CREB, which is the transcription factor. It phosphodilates the CREB, which means it adds phosphate group to the CREB and binds to the gene, and the CREB binds to the gene, and transcription of protein thus takes place. Okay, consider this figure. This is the G protein which is attached to the cytoplasm, the C terminal of the serpentine receptor, which is the G protein coupled receptor. The alpha subunit then dissociates when a ligand binds to the receptor, and thus the ATP and thus the GDP is released and GTP comes and bind here. Dissociation of the alpha from the G protein activates the G protein, and the alpha binds to the adenyl cyclase. The alpha dissociates and binds to the adenyl cyclase, and thus it converts the CAMP. It converts the ATP into CAMP. The CAMP further activates the protein kinase A, which has 2 regulatory subunit and 2 catalytic subunit. CAMP binds to the regulatory subunit of the protein kinase A. And this the regulatory subunit of the protein kinase A goes to the nucleus. Inside the nucleus binds with the CREB, which is a transcription factor, and it phosphodilates CREB, bind to gene, and transcription of protein takes place. Here cellular response takes place.